soft corals are associated with a variety of bacteria, and bioactive chemicals produced by these bacteria have been found in various marine settings across the world. Its substantial use as antifungal agents is shown by the coral-associated bacterium's strong bioactivity, specifically against biofilm-forming bacteria. This study was aimed to understand the anti-biofilm activity of endophytic bacteria associated with Red Sea soft corals against primary fouling bacteria. In this study, endophytic marine bacteria isolated from Red Sea soft corals were exposed to the extraction of secondary metabolites and evaluation of the extracts for the anti-biofilm activity to identify chemicals that can inhibit the colonization of surfaces by biofilm-forming bacteria. 6 bacteria were isolated from 2 species of soft coral, which are Sarcophytum glaucum and Heteroxyna persistens. An effective method for preventing biofouling on submerged structures is to eliminate early surface colonizers.
The first method is collection of soft coral. The first procedure is sample collection for Upper Creek of the Central Red Sea coast. There will be by using hand through scuba diving. Secondly, the sample divided into 3 parts. First part, processed immediately. Second part, stored in 20 degrees Celsius. And lastly, they will be preserved in 70% of ethanol. Next, extract will be concentrated using a rotary evaporator. And next, crude extract will solubilize in DMSO and stored. Lastly, mass culture in 2 liter flask for subsequent analysis. The second method is isolation and endophytic bacteria. So the next procedure is they will be washed many times with 0.22 microliter with the filtered seawater FSW to remove debris and microbes or planktons that will loosely attach. Next is aseptically cut into small pieces and transferred into sterile flasks containing sterile FSW. They will be shaken vigorously. Next, there will be done seal dilution. Lastly, inoculate media with soluble marine agar and marine nutrient agar using spread method. The third method is isolation of genomic DNA, PCR amplification, and sequencing. Genomic DNA from each of the bacterial strain was extracted by first harvesting in the bacterial cells through centrifugation for 10 minutes at 500 grams. Secondly, blood search limited to the type material option from the National Centre for Biotechnology Information (NCBI) was used to assign type strain with the highest blood score and nearest match found in GenBank. So these two will be used for comparison. The fourth method is phylogenetic analysis. First procedure is phylogenetic pipeline was used for the construction of the phylogenetic tree using the one-click mode option. Secondly, curate the alignment with G-blocks. Thirdly, build tree with maximum likelihood method using PyMime. The next one is the pipeline finally draw the tree with tree die. Lastly, the result was exported as numeric format to the ITOL web server for annotations and evaluation of the tree.
Next, the extraction of secondary metabolites was carried out with the organisms that were first screened for activity against biofilm bacteria and will be cultured in marine nutrient broth overnight. The supernatant and residue were recovered after centrifugation. Ethyl acetate was used as a solvent for culture supernatant extraction, while methanol was used as a solvent for whole cell extract. Solvent phase was separated and the methanol was added to the residue and they both filtered. Extracts were then concentrated using a rotary evaporator and the crude extracts were solubilized in dimethyl sulfoxide and stored. After that, paper disc diffusion method was used to test antibacterial activity. Crude extracts were tested against 3 biofilm-forming bacteria which were previously isolated from aquaculture cage nets in the Red Sea. The selected bacterial strains were known for their prevalence in microfouling. Previous studies confirmed their biofilm-forming ability and barnacle-level settlement-inducing capability. Then, the bacterial cultures were standardized to Macfarlane standard. Extracts were applied to discs and placed on inoculated plates with the positive and negative controls. The next step was conducted by micro-titering biofilm inhibition assay to test the extract's ability to inhibit or reduce biofilm formation. Planktonic stage and biofilm formation were evaluated using a spectrophotometric assay. Absorbance at OD 630 was measured to estimate biofilm inhibition. Crystal violet assay confirmed the biofilm forming capability of bacterial strains. 3 strains representing specific bacteria were used and the sterilized microplates were conditioned with autoclave FSW. Bacterial cultures were diluted and added to the plates along with the extracts. Plates were then incubated and the pentonic growth inhibition was measured. The unattached cells were removed by washing and attached cells were stained with crystal violet. Excess stain was removed and the remaining stain was solubilized. Lastly, the metabloid analysis of the extracts was performed using gas chromatography mass spectrophotometer. The column temperature was programmed with specific temperature settings. Helium was used as the carrier gas and the GC injector and GC-MS interface were maintained at specific temperatures. Possible metabolites were identified based on their molecular ions and compared with the library database. Chemical structures of bioactive compounds were drawn using ChemDraw.
In this article, the endophytic bacteria isolated from soft coral were identified based on several factors, which are their colony morphology, cell morphology, biochemical characteristics, and 16S RNA sequencing. Based on their morphological features, the results revealed that all of the isolated strains, excluding IAC4, belong to the family Bacilliaceae. This can be shown by their colony morphology, which mostly resembles the Bacillus colony, which is moist and has a smooth texture. Compared to colony morphology of IAC4, they will form a dry colony which is similar to Oceanobacillus colony. The most important method that is used in identifying the members of Bacillus group is by using 16S RNA gene similarity. According to the analysis, it is found that all the isolates from soft coral are revealed to belong to Bacillus genus with the tight evolutionary relationship. This is due to the fact that they have the highest percent of similarity with the family Bacilliaceae. For example, IAB1, IAB2, and IAC2 strains have the highest similarity with Bacillus subtilis. As for IAB5 and IAC1, they recorded the highest percent similarity, which is about 97.56% with other members of Bacillus cereus group. From the percentage similarity, it can be concluded that the highest percentage of similarity will show that the genes of those bacteria are similar to each other. Therefore, they are classified under one group. In terms of the biochemical test, the strains that are similar to Bacillus genus will have a positive result for catalase and negative result for oxidase test. This is because Bacillus genus does not have the cytochrome C oxidase that oxidizes the test reagent. However, IAC4 strain, which is considered to be similar to Oceanobacillus, will provide positive results for both tests. According to the result of antimicrobial activity of the extract, strain IAB5 recorded the highest zone of inhibition for both extract of culture supernatant and whole cell extract, which is about 26 mm. As for strain IAB2, both of the extract are found to have 100% antibacterial activity against all the biofilm-forming bacteria compared to other strains. Among all the biofilm-forming bacteria, Pseudo-Ultramonas schioia-sacensis is reported to be more susceptible towards all culture supernatant extract from all isolated strains. As for gram-negative bacteria such as P. schioia-sacensis and Vibrio harveyi, their susceptibility depends on their membrane permeability, which provides extra protections against the entry of antimicrobial compounds. In this case, they are found to be more susceptible due to the destruction of their outer membrane by the extracts. This will then lead to the exposure of the underlying peptidoglycan layer and allow the penetration of extract through the bacterial membrane.
Due to the antibacterial activity recorded by the extracts against the biofilm-forming bacteria using the diffusion method, each of the extract was evaluated for anti-biofilm formation activity. Based on the result obtained from the article, extracts that were obtained have shown positive outcomes in biofilm formation. The bacteria population's capacity to adhere to surfaces plays a crucial role in the initial development of biofilms. In addition, antimicrobials tend to be more resistant to bacteria in biofilms than they are in planktonic form. Therefore, some extracts exhibit antibacterial or antibiofilm properties that can be attributed to the bioactive compounds present in the extract. Results show the percentage of biofilm activity of the extract in whole cell and culture supernatant. As we can see from the first figure, the highest planktonic growth inhibition activity was 89.9% by the whole cell extract of strain C4 against P. schioia-sacensis. This is due to extracts with both antibacterial and antibiofilm properties may have interfered with the biofilm strain's ability to detect forums or with the breakdown of EPS and the creation of cell walls, which can be thought of as potential sources of antibiofilm chemicals. The second figure shows the ratio of the absorbance of the biofilm or planktonic at 630 nanometer shows that most of the extracts have reduced the biofilm bacteria into poor biofilm formers when compared with the control. This study indicates that by drastically lowering the biofilm mass of the tested organisms when compared to the control, the majority of the extracts have antibiofilm activity. In the table shown, it is a list of the compounds detected in both species of soft corals by this method. However, the most observed compounds found in the extracts are fatty acid derivatives and nitrogen-containing compounds. For example, compound that is found like the one in the slide have the highest frequency of occurrence in most samples. This compound shows that it has both antimicrobial and antioxidative properties. This suggests that the substance may have particular antibacterial and antifoaming qualities. And there are also other compounds as stated in the table that are extracted from the extracts in the present study.
In conclusion, the endophyte extracts significantly inhibit the growth of the tested microorganisms. The extract's ability to prevent biofilm formation varies from 3.16 to 98.41%. The presence of certain substances in the extracts reveals the bacteria's capacity to create substances important for preventing the formation of biofilm on surfaces, which might be used as a novel source of antifouling substances. Extracts from endophyte found in soft corals were found to be effective antibiofilm agents against bacteria in biofilms that may produce bioactive secondary metabolites.